Right. Good morning, good morning, good morning. Okay, so we are going to continue talking about mutations. We're going to continue working on the worksheet. Let me see if mine will come up this time. Yay! All right. And if I'm not mistaken, we were right about here. All right, so take a look at that. I'm gonna wait for everybody to come in. If you haven't filled in the sickle cell part of it, please do that. I'll wait a few minutes before I start. Let's see if everybody's coming in. Might have one or two people went on. There's a field trip today, so might be missing some folks. Why is this person coming back to this class? I have this person trying their hardest to get into class, and they're not in this class. But for some reason, it won't let him in, so I can tell him he's trying to get into the wrong class. All right, so again, hopefully you filled that part in. We're going to try our very best to get through this worksheet today. Remember, you need to have this chart handy somewhere. Try to reproduce it so that you can have a side by side version. So because you have to keep referring to the chart, so it would be helpful if you could put it where um, you can see it. The other thing I want you to, oh, that's right. I was just about to say I want you to. We'll come back to that in a sec. I want you to go ahead and get ahead of this benchmark test because you're Georgia milestone subject. The school has created benchmark exams. So you will have a benchmark test as soon as we come back. Hold on. And I'll remind you again on Friday. All right, benchmarks. Benchmarks have to be the first week in December. So we're off next week. So when you come back, I will see you on Tuesday and there will be a benchmark. So let me write that down that yours is on Tuesday, December, what period? December 2nd. You need to make a note of that as well. All right, so December 2nd, you are going to do a benchmark exam. This is an exam that they have created in progress learning. And so it'll be administered there. So you'll come in, I'll give you code to get into that exam on that day. What a benchmark test is it covers everything that the course is supposed to be about. So there are things that we've done, and there might be a couple of questions on things that we haven't done yet. And bench, it's coming on December 2nd. So write that down somewhere, December 2nd. So this is your calendar. I'm on your homepage. Yes. All right. So December 2nd. That's the benchmark. And again, what a benchmark test is it covers everything that's supposed to be in the course. And it is information that the school uses in order to place you in your next class. They use it to determine whether you need extra help because they're going to try to make sure that everybody gets all the help that they need to pass the milestone test. Okay, so it identifies your areas of strength and the areas of weakness. So you need to take this test with the attitude that I'm going to do my very, very best because it doesn't count for a grade, it counts towards placement. Okay, so you need to take this test seriously. And because of the benchmark test, we're not going to have a end of term exam. We will simply have a end of unit exam, and it will probably be December the 12th. Okay, so end of unit for your class is December 12th. So that'll only be on the unit that we're on now. Okay, all right, everybody clear? All right, so like I said, I'm on your homepage on the calendar on your slideshow that has the exam schedule. So the first week in December is for benchmarks. This is something that you're going to take in progress learning, and this is information the school needs to place you in your class or give you some help. They're going to send the information back from me, and I'm going to know what your areas of strength and your areas of weakness are. Okay. All right. So benchmark December 2nd, end of unit test on December the 12th. Alrighty, so let's go back to the module. Any questions, any questions, any questions? Okay, because Friday is the last time I see you, then we're off for a week, and then you come back, and you'll be coming back on a Tuesday and taking the tests on that Tuesday. We need to do it Tuesday in case you need more time, because then you'll be able to finish it on Wednesday if necessary. Hopefully that won't be necessary. You guys are good at taking tests. So I believe that you'll get it all done. During the first go round. All right, so we're back to our worksheet. And again, I'm walking through this with you, so you need to actually just give me 1 minute. I'm going to stop sharing and do attendance. And I'll be right back. But while I'm taking attendance, you make sure that you are at least up to sickle cell anemia. Then we're going to do frame shift. So these are types of mutations. Okay, so we did the first one with the cow and the human. That was about a point mutation. And we have decided that insulin is a point mutation. It is not a silent mutation. Silent mutations mean that it has no effect. Point mutations means that it's going to cause a problem. In this case, the mutation will cause the inability to produce insulin, so you'll have diabetes. Okay, what you want to do is keep track of what type of mutations are cause which diseases, because that's a test question. All right. Okay, and I had already stopped sharing, so hopefully you were paying attention to what I just said. But I'll come back and repeat it as soon as I finish attendance. 'Cause All right, Avonay, you should be on camera. Okay, we got one on the field trip. Okay, so it looks like everybody who's out is on the field trip. Mm, mm. All righty. Right. Oh, what I started to say before all of that. Okay. Okay, what I started to say, which led me to drift towards uh, um, the benchmark test, is that at the end of this unit, there is a study guide. And we've downloaded other study guides before. All right, this one. So I want you to go ahead and download this so that you can be looking at this over the break. And that will help you with the questions about DNA synthesis. Um, I might even go over some of this on Friday when I see you. So go ahead and download this and that'll help at least with that. So we have done okay. So that's at the end of this unit. So our unit is DNA. And again, the test on DNA will be December 12th. The benchmark um, covers everything that's all right. So far, so far we have done scientific method, classification of living things. We've done macromolecules, cell structure, cell energetics, and DNA synthesis. So that's what we've done. So it is expected that you can pull up the review pages for all of those, and that'll help you prepare for things we've already done. Then things we haven't done that they're going to test that you might run into are things like mitosis and meiosis. Now, she mentioned that in the video, but we still haven't done it yet. So think they will ask questions about genetics and ecology and taxonomy, but don't worry about do the best you can with those kind of questions. Benchmark tests are always about trying to see what you know from what you don't know. Okay, so they know there's going to be some things that you don't know, and that's expected. All right, but right now, it's expected that you know everything up to DNA. Okay. All right. So again, download this review page. I think what I'm going to do on Friday is just do that page with you. Start working on it so that you at least have that review under your belt in preparation for the benchmark. So let me put that on my calendar. And you can go back and review all of the other ones. So pick a day when you have nothing to do during the break and review those other topics that you have access to. Um, a review page. Every unit has a review page. Some of them we've done already, so you might just have to download it and see what you've done. And I think all of them did. All right. Yep. This one had a review page. So they all have a review page. Yep. So that's on the first one, scientific method. So again, they all have a review page at the end. There's that one, and we had to turn that one in. So that one you definitely already did. We did. Oh, there it is. I have too many other things on this page. All right, so we did that one. We looked at that one. You didn't have to turn it in. Yes, you did. And then now I'm telling you to work on this one. All right, so that being said, let's get back to the worksheet. How much time we got? We got a few. All right, so again, we're looking at why didn't come up like that the last time. Okay, comes up different every time I open it. All right, so we are here. So we had decided that, again, diabetes is a point mutation because it causes a change in the protein that is produced, therefore it causes a disease. So that's a bad mutation. Some mutations are good, some mutations are bad. All right, let's look at, hopefully you've gone through and you filled this chart in, and you're not just sitting back waiting for somebody to do it for you. All right, so you should have completed the row for RNA. You put in the triplet codes. So you should be checking your page, and I'm going to read it up because we're running short on time, and you all take forever to answer me. All right, so the first one should be CUG, and the amino acid that you should get from that is LEU. Now remember that these amino acids are abbreviations. Those are not triplet codes. That's actually the shortened version of the name of the amino acids because they're long. So they give you a three-letter abbreviation. The triplet code for TGA is ACU, and that's going to give you thurine, which is THR. The triplet code for GGA should be CCU, and that'll give you proline. And then the triplet code for CTT is GAA, and that'll give you GLU. And then finally, the triplet code for CTC is GAG, and that will give you GLU also. So notice you get two different ways to get GLU. All right, so that's the way it's supposed to look. And again, for sickle cell anemia, if you're looking at the picture up there, and let me make mine bigger. All right, so these are these cells up here. Nice round donut looking one is what a normal red blood cell should look like. And it's the hemoglobin that is the chemical inside the red blood cell that makes it red. And its job is to carry oxygen. When it is not shaped properly, it doesn't carry as much hemoglobin, so it's not carrying as much oxygen. So people with sickle cell tend to be short of breath. Some of them have to have oxygen tanks, and sometimes their life expectancy isn't very long because of it. Okay. There is no cure for this. You simply have to learn to deal with it. All right, so the next row is what the blood cell will look like if you have your protein strand will look like if you have sickle cell. All right, so the first one is the same as the one above. They're both GAC, so they're both going to produce leucine. The second one is also the same as the one above, so you should repeat that information in that box as well. Both of them have GGA, so you should have the same information in that box as well. Here's where we have the change. The first one in the normal one is CTT, and this one is CAT. So somebody tell me what you're going to get. What is the triplet code and what is the amino acid that you're going to get for this one? And by the way, the last one is the same as above, so you just feel that both of those should be our difference. So our difference is in this one triplet code. What should go here? Some wonderful person, go ahead and tell me in the chat. What will be the triplet code? How do we translate CAT, and what amino acid is that going to bring? Okay, thank you, Brooklyn. And she said GUA, and what amino acid is that going to bring? Valine. Very good, very good. All right, so everybody should have that box filled in. All right, so because this is a single gene or a single amino acid change, is it going to be a point mutation or is it going to be a silent mutation? That's the question they're asking you here. This is definitely going to cause sickle cell because it's different. They're different. So what's the word we want to write here? It says, what type of mutation is shown? Somebody, anybody? You have a choice between point or silent. Is it going to be a point mutation or a silent mutation? Raymond, point mutation or silent mutation? Everybody's waiting for somebody else to answer the question. Raymond, what you think?
The silent mutation is
All right, remember up here, silent mutation means there's no functional difference. Functional difference. There could be an amino acid difference, but it creates the same amino acid. I mean, it could be a different RNA sequence, but if it produces the same amino acid, then it is okay. This is not okay. It creates a different amino acid. You now have valine instead of glucine. So this is going, folks, this is going to be a point mutation. Okay, point mutation. The shape of the protein changes and becomes less functional or not functional at all, which is the type of diet. With diabetes, it becomes different. Sorry, I'm trying to stay right there. All right, so that's what we have here. We have something that has changed the function of the protein. So it is a point mutation. So right now, so now we have diabetes is point and sickle cell is a point mutation. All right, make note of that. All right, we're going to, and I'm going to let y'all work on this, but I'm going to explain what we're doing here. All right, so we have a different type of mutation now, frame shift. So what we're looking at is, right now we've inserted uh, a nucleotide. Each of these is a nucleotide. So if you look at this one up here, you want to do the same thing. You want to look at the triplets. You want to create an RNA strand and then find the amino acid. What is happening with the mutant is they've inserted an extra nucleotide. So they've highlighted that A, and it shifted the whole line over. Okay, so now when you're reading it in groups of 3, it's a shift. So then because you're reading it in groups of 3, this, instead of it being AGT, now it's AAG. So what amino acid are you going to get there? And then answer that question. Okay, then they're telling you cystic fibrosis is a disease that is caused by a frame shift mutation. Okay, then they have another one, muscular dystrophy. Okay, so they want you to look at this one as well. All right, and I'm gonna let you try to get at least those 2 done before the end of class. And that just leaves us with 1 page to go. All right, so your objective is to finish this worksheet, but at least try to get, does it always have to be? Yes, it usually is. Sometimes sometimes it can be more than one because you could have a whole section missing. So in which case you would be leaving out a whole amino acid. But it is usually just one nucleotide causing the problem. And again, that's the problem comes because we read it in sets of 3. So if you change the arrangement of the sets of 3, it now codes. It's still going to read what's there, but it's going to read for something that's not supposed to be there. Okay? Good question. All right, so I'm going to let you all work on that. We have quite a few people absent, so you all will be the geniuses that help me answer it for everybody else next class. And if you have a question while you're working on it, please ask. Please let me know. So as you see, we have a couple of diseases that are frame shift. The DNA codon chart is at the end of the worksheet, but it's also um, it's at the end of the worksheet. So there it is. That's your codon chart. But it's also, and the reason I like the one in your module. Okay, why is this not letting? Okay. The one in your module has the names written out. So it's kind of like that one. Um, where's the go back button? Okay. Like I said, that just, oh, I know, because I'm looking at that as an assignment. So it's not the module. Come on. All right, so if you look at the unit on mutations, I don't know why I didn't get it. These are the codon tables as well. Okay. And I also have them as pictures when we did DNA replication. There. All right, so there's another one. Okay. So they're all over the place. All right, if you have no other questions, folks, we're good for today till I see you on Friday. So that codon chart is in a couple of places.